Hello everybody and welcome back to biosc140. This is part 4 for membrane dynamics. So in the past videos, we've talked about movement across a membrane. So compartments are important in your body because how many chemical reactions take place in your body? A lot. A lot of chemical reactions take place in your body, and chemical reactions need very specific environments in order for them to happen properly. Now, just one specific environment is not, all the reactions needed for life don't take place in just one compartment. So you have lots of compartments, and they each have their own unique environment inside to allow their own unique chemical reactions to take place. And in order for it all to work in one system, you need to be able to move things in and out of those compartments. So we've been talking about moving things in and out of these compartments, moving things across membranes. And up to this point, we've only talked about passive mechanisms. And by passive, we mean mechanisms that do not require the input of energy, the use of ATP. With passive, there is a concentration gradient that's already set up, and the energy comes from that already set up concentration gradient. In this lecture, in this part, we're going to talk about active transport. So in active transport, you go away from equilibrium. You create disequilibrium with active transport. You move things from low concentration to high concentration. You move things against their concentration gradient. This requires energy. When we say requires energy, it requires the use of ATP. We'll talk a lot about ATP in future lectures. There's two primary types of active transport. There's primary active transport, which is the direct use of ATP. And by primary, we mean you want to move the orange square across the membrane. Well, orange square binds the transport protein, ATP binds the transport protein, and bam, it moves across. ATP binds to the protein, the transport protein, that is doing the transporting directly right when the transport is being done. Secondary or indirect active transport is when you have a primary active transport set up a chemical disequilibrium, set up a situation where you have high concentration on one side of the membrane, and you use that concentration gradient that is set up by primary active transport to power another transport. So look at this. Active transport, primary transport, moves silver circle into the extracellular fluid, and we have high concentration of silver circle in the extracellular fluid. Disaccharide, green disaccharide here, we have high concentration of green disaccharide inside the cell, low concentration outside. So the concentration gradient for silver circle wants to move into the cell. The concentration gradient for green disaccharide wants to move out of the cell. But we want to move green disaccharide into the cell against its concentration gradient. So we're going to couple these two movements together. Silver circle wants to move into the cell, so we're going to have silver circle move down its concentration gradient, and at the same time, when silver circle moves down the concentration gradient, it's going to bring green disaccharide with it into the cell. It's the use of a gradient established by primary active transport to move it in. There is energy stored in concentration gradients. You're using that stored energy in concentration gradients to move green disaccharide against its concentration gradient. It requires energy. It moves green disaccharide from low concentration to high concentration. It creates disequilibrium. Now, we're going to talk about this a lot with with nerve cells. With nerve cells, we're going to talk a lot about the sodium-potassium pump, the sodium-potassium ATPase, so it uses ATP pump. This pump creates chemical disequilibrium within a nerve cell, which allows the nerve cell to function properly and send those electrical signals. It uses ATP and it sends 3 sodiums to the outside of the cell where we have high sodium concentration. And at the same time, it sends 2 potassiums into the cell where we have high potassium concentration. So it's moving potassium and sodium against its concentration gradients, creating this chemical disequilibrium. It directly uses ATP to pump 3 sodiums out and 2 potassiums in. Right here, we have another visualization of it. Primary active transport directly uses energy stored in ATP. It's the binding of ATP that causes the conformation change, that causes the shape change, which transports the 3 sodiums out and then the 2 potassiums in. And when we do this, we're moving 3 positive charges out and 2 positive charges in. So that gives us a net loss of charge within the cell. You move 3 positive charges out, you move 2 positive charges in. That gives us a net loss of charge within the cell. And this creates an electrical and chemical disequilibrium in the nerve cell. We're going to go into this in a lot more detail when we talk about exactly how nerve cells function. But if you go back a couple slides, you can see that we have high concentration of sodium outside, high concentration of potassium inside. That is a chemical disequilibrium. The concentrations of the chemicals are in disequilibrium. Now, if you look at just the charges, we have more negative charges on the inside, more positive charges on the outside. We're moving 3 positive charges out and 2 positive charges in. We have negative charge on the inside. That's an electrical disequilibrium. So the sodium-potassium pump creates electrical and chemical disequilibrium on the nerve cell. So as secondary active transport, you couple the movement of a molecule moving down its concentration gradient to push another against its concentration gradient. There is energy stored in concentration gradients. Concentration gradients are potential energy. For anyone who's taken a physics class, there is energy stored. It's like a battery in chemical, in concentration gradients. And secondary active transport uses that energy stored in a concentration gradient to move one molecule against its concentration gradient. It relies on disequilibrium generated by primary active transport. Now, there's a few flavors of secondary active transport. So co-transport's the transport of more than one molecule simultaneously, so like the sodium-potassium pump, co-transport. With a symport, it's in the same direction. With an antiport, it's in opposite direction. So sodium-potassium pump, how would you classify the sodium-potassium pump? Antiport. How would you classify this situation with silver circle and green disaccharide? Symport, they're both moving in the same direction. They're both moving in the same direction. It's easy to write test questions on vocab words. Pay attention to all underlined words. So there's one more main type or big type of active transport. So endocytosis. Endocytosis brings substances into the cell by expending energy. So endocytosis is the process of capturing a substance or particle from outside the cell by engulfing it with the cell membrane and bringing it into the cell. It's kind of like when the cell like amoebas around something and brings it in. You know, I've used that term in the past, like amoebaing, kind of just absorbing something in your membrane and bringing it in. That's what endocytosis is. And there's 3 different flavors, 3 different types of endocytosis. You've got phagocytosis. So this root right here, That means to eat. So this is when the cell eats. It's the cell membrane engulfs a substance to be absorbed and then buds off to form this vesicle. So we have this substance right here that the cell wants to eat, and you can see how the membrane kind of wraps around that substance. The membrane wraps around that substance until it's completely wrapped up, and then it brings it inside this vesicle. With pinocytosis, the cell engulfs fluid. So it's the same basic process, but it's engulfing a fluid and not necessarily a specific substance. With receptor-mediated endocytosis, it is an extremely selective process of importing materials to the cell. This specificity is mediated by receptors, which will take the entire complex into the cell. So the main difference between phagocytosis and receptor-mediated endocytosis is phagocytosis, the cell just kind of goes out and around an object, a substance, and brings it in. There's not really a very specific process. With receptor-mediated, you have something bind to a specific receptor on the cell membrane, and that's what starts the process. With phagocytosis, there's no specific receptor binding that starts the process. Receptor-mediated, a specific binding starts the process. And for example, some viruses will bind to a specific receptor on a cell, which leads to the cell to bring it in and let it through its defenses, through its membrane, and into its cell body. So receptor-mediated is very specific. With exocytosis, it's kind of the opposite. With exocytosis is the process of a vessel fusing with the plasma membrane. So these vessels are made up of phospholipid bilayers. So a phospholipid bilayer vessel or capsule within the cell fuses with the cell wall, the cell membrane, and releases its contents into the extracellular space. All right, that's the end of membrane dynamics lecture. Email me if you have any questions. Have a fabulous day, everybody.